Okay, students, welcome to the next session, that is NCERT First PUC Syllabus Biomolecules Chapter 9. In this session, we will discuss few bits. The first bit, which of the following is not strictly a biomacromolecule? Proteins, lipids, polysaccharides, and nucleic acids. In these three, which is not a strictly biomacromolecules? Generally, when we will grind the living tissue, when we will filter the living tissue after grinding, if you filter, two fractions are formed. This is called acid insoluble fraction. Insoluble fraction. It is called acid soluble fraction. Acid soluble fraction contains amino acids. Its molecular weight is 18 to 800 Daltons. So acid insoluble fraction, its molecular weight. So generally acid insoluble fraction so contains proteins, nucleic acids, polysaccharides, and lipids. So in this acid insoluble fraction, this molecular weight is 10,000 Daltons will be there, except lipids molecular weight. The lipids molecular weight is 800 Daltons. So the lipids are not considered as a strictly biomacromolecules. It comes under acid insoluble fraction. Okay, so because the molecular weight of lipids does not exceed 800 Daltons, but they come under the acid insoluble fraction. So that in this question, the correct option is lipids. Okay, so the correct option is 2. Okay, remaining proteins, polysaccharides, nucleic acids, they comes under biomacromolecules. Okay, that's all. Next question, second question. A secondary metabolite that act as a toxin. Which of these? So generally here 4 options given. Among those 4 options, one is secondary metabolite, that is carotenoids. Carotenoids. What is that? Carotenoid is a pigment. It does not act as a toxin. Curcumin. Curcumin. It is a drug. It does not act as a toxin. So abrin. Abrin. It is an extremely toxic toxalbumin substance. It is found in the seeds of rosary pea. So abrin, rosary pea seeds, this kind of a toxin is found. Okay, so it is found in Florida. Florida, it is found in Florida. Okay, so it is a toxin. Abrin is a toxin. Next, monoterpenes. Actually, monoterpenes. Okay, it is a terpenoids. 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 It is not a toxin. Here, the second option among 4 options, third option, abrin is correct option. Correct answer. Abrin is correct answer. Next, come to third question. A secondary metabolite that is alkaloid in nature. Secondary metabolite that is alkaloid in nature. See here, which of the following is an secondary metabolite that acts as an alkaloid nature? Codeine. 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 It is a secondary metabolite that acts as an alkaloid in nature. It treats pain, coughing, and diarrhea. So that it is used as an alkaloid codeine. Next, anthocyanin. Anthocyanin, it is a pigment. Okay, it is not an alkaloid. Gum. Gum is a polymeric substance. It is not an alkaloid. Abrin, it is not an alkaloid. It is a toxin. About which we discussed. Toxin. Okay, so that here among these 4, third question correct option is 1. The option 1 is correct. Next, come to the fourth question. Peptidoglycan present in bacterial cell envelope is. So generally here if you see bacterial cell envelope like this way, cell outermost part is called like this way, cell envelope. This is the cell envelope. So this cell envelope is made up of by peptidoglycan, peptidoglycan. It is present in the bacterial cell envelope. This is called cell envelope. Cell envelope. So in this cell envelope, whatever may be the peptide glycan is present, what is that component? What kind of a components are present in that one? Okay, see here, made up of cellulose. Actually, in this bacteria, cellulose only not present. So option 1 is wrong. It is a heteropolymer. Yes, sir. Actually, peptidoglycan is a heteropolysaccharide. Heteropolysaccharide made up of two alternate amino sugars. Two alternate amino sugars. That is N-acetyl. That is N-acetyl glucosamine. N-acetyl glucosamine and N-acetyl muramic acid. These two sugar amino acids are present in the peptide glycan, so that it is a different means different polymer, heteropolysaccharide. Okay, that's all. So fourth option, the correct answer is 2. Fourth option, the correct answer is 2. Next, come to third. It is an oligosaccharide. So it is the wrong option. Actually, oligosaccharides in peptidoglycan is not present. It is a homopolymer. It is not a homopolymer. It is a different polymer. Different polymer. So that fourth question, the correct option is 2. Next bit. Fifth question. In glycine, the R group is replaced by, in the glycine, the R group is replaced by, in glycine, the R group is replaced by which of these? Generally, a methyl group. So first of all, amino acid group we have to draw. NH2, CH, COOH, R. This is R is alkyl group. The general formula for amino acids is like this way. In the R group, glycine. Okay, so in glycine, the R group is replaced by, this R group is replaced by H. How it is? NH2, CH, COOH. It is H. When R group is replaced by H, then it is called glycine. Okay, so in glycine, the R group is replaced by hydroxy. R group is replaced by hydrogen. Sorry. First one is methyl group. It is wrong option. If methyl group is added, then it is alanine. If hydroxymethyl added, then it is serine. If carboxylic group is added, then it is another group. Carboxylic group added, then it is another group. Okay, so if hydrogen is added in glycine, the R group is replaced by hydrogen. So in this R group, hydrogen is added. That's why it is called glycine. So fifth question, the correct option is 4. 4 is correct option. Okay, so glycine is simplest amino acids in which R group is replaced by hydrogen. Next, sixth question. Zwitterions are ionized species of. See, what is that? Ionized species of? Is it amino acids? Is it acidic amino acids? Basic amino acids? Neutral amino acids? Yes, all of these. Okay, when the zwitterions, generally zwitterions, ion having separate positively and negatively charged groups, positive group and a negative group. Okay, so basic amino acid group will be there, acidic amino acid group will be there, neutral amino acid group is there. Okay, Since amino acids can carry both positive and negative charges simultaneously, hence such type of groups are termed as deuterions. So that for the sixth question, correct option is 4. Fourth option. Okay. The most abundant protein in animal world is, in the animal world, chitin. Chitin is a polysaccharide. Collagen is a protein. Peptidic glycan is a polysaccharide. Hyaluronic acid is a polysaccharide. So among these four options, collagen is the abundant protein in the animal world. So that eighth question correct option is 2. Next, ninth question. Proteins which catalyze biochemical reactions in the living world, those are known as generally proteins. See, enzymes are proteins. Those catalyze all the biochemical reactions in the living world. So ninth question, option 1 is correct. Let's see hormones. Generally, hormones can be steroid and amino acid derivatives. These are not involved in biochemical reactions. Antibodies, generally antibodies are glycoproteins. These are not involved in biochemical reactions. Receptors, generally receptors are associated with sensory reception. Therefore, among the four options, first option is correct. Next, the amino acids in a protein are held together by, generally, amino acids in a protein chain held together by peptide bond. Peptide bond. See, here four options. Glycosidic bond, it is not located in between the amino acids in a protein. Phosphodiester bond, it is not. Hydrogen bond, it is not. Peptide bond. See, first of all, what about glycosidic bond? Glycosidic bond is formed between the two monosaccharides, that is, two sugar molecules. Okay, so that is called glycosidic bond. Phosphodiester bond, this bond is formed in a nucleic acid, that is DNA or RNA between the phosphate and hydroxyl group of a sugar. That is called phosphodiester bond. So that it is wrong option according to the question. So peptide bond is formed between the two amino acids. So it is one amino acid, it is one amino acid. So this is the amino acids. So COOH group of one amino acid, COOH group of one amino acid is reacted with amine group of another amino acid. Okay, so COOH group of one amino acid reacted with the amine group of another amino acid and releases water molecule, and here formed peptide bond. Peptide bond. So that third option is correct option. Okay, that's all. Next, here 11th question. A protein which exhibits printed structure, that is called, okay, here four options given. Among these four options, one is protein that will exhibit pleated structure. Pleated structure means what? Like spiral-like structures. Which is that? Okay, so fibroin, hemoglobin, enzyme, and alpha keratin. It is the symbol. This is alpha keratin. So here also alpha pleated structure. Okay, so here misprint alpha pleated structure. So among these, which one? Okay, generally fibroin. Okay, a protein. This is a protein which exhibits alpha pleated structure. So alpha, like this, alpha pleated structures in the proteins. Okay, that's all. Option 1 is correct. Let's see, hemoglobin also protein only. Enzyme also protein only. Keratin also protein only. So hemoglobin generally exhibit quaternary structure. Okay, so that it is a wrong option. Generally, enzymes are tertiary structures. Okay, keratin, alpha keratin, it is secondary structures. Okay, alpha means what here? Secondary structure. A protein which exhibits secondary structure, that is fibroin, which exhibits secondary structure. So 11th question, the correct option is 1. Next bit, an example of protein with quaternary structure is here. 4 examples of protein with quaternary structure is myoglobin, hemoglobin, keratin, and all of these. Already here we have seen myoglobin is a protein. It is a tertiary structure. Protein at the tertiary structure acts as a myoglobin. Hemoglobin, it is quaternary structure. At quaternary structure, protein acts as a hemoglobin. Keratin, it is secondary structure. Okay, so an example of protein with quaternary structure is hemoglobin. 12th question, correct option is 2, hemoglobin. Next, lecithin. Lecithin is, okay, so type of wax. Actually, lecithin is not a wax. Okay, it is wrong option. Okay, so phospholipids, actually, lecithin is a phospholipid. Okay, lecithin is made up of molecules of glycerol, phosphate group, 2 fatty acid molecules. Okay, so that is called lecithin. Lecithin is a phospholipid which contains glycerols and phosphates, mainly glycerol and phosphates. So that 13th question, the correct option is 2. Okay, that's all. Yes. Remaining 3rd and 4th options are wrong. Okay, so here lecithin is phospholipid which contains mainly glycerol and phosphates. That's why second option is correct. Remaining, it's not a type of fats, it is not a oil, it's not a fatty acid. So 13th question, correct option is 2. Next, 14th question. Lipids that insulate the nerve fibers are, okay, what kind of a lipid that insulate the nerve fiber? See, nerve fiber means what? So see, it is axon. From the cell body, this is called cell body. It is the structure of nerve. Here, these are what? Dendrites. Dendrites. Okay. So from the cell body, there is long tube-like structure that is called axon. So the axon is covered by nerve fibers. So the lipids that insulate the nerve fibers. What kind of the lipid? Is it lecithin? So lecithin is wrong. Lecithin is phospholipid. Okay, lecithin is wrong. Cholesterol, it is fat. It is not. Okay, suberin. So it is also, it's not, actually it is located in the cell wall. In the animal cells, suberin is absent. Glycolipids. The glycolipids are present in myelin sheet of nerve cells. So nerve cells are 2 types: myelinated neurons, non-myelinated neurons. In myelinated neurons, so the glycolipids acts as insulating glycolipids in myelinated neurons. Therefore, for this 14th question, correct option is 4. The pentose sugar present in RNA is. The pentose sugar present in RNA is. See, pentose sugar. Galactose, yes, it is wrong option. Galactose is not located in RNA. Sucrose, it is not located in RNA. Ribose, it is located in RNA. Ribose is a monosaccharide sugar present in RNA. Okay, so that 15th question, correct option is 3. 4th option is fructose. It is not located in pentose sugar. Okay, that's the correct option is 3. Next, nucleoside is. nucleoside. In this nucleoside, sugar plus nitrogenous base is called nucleoside. One is okay. Sugar plus phosphate. Sugar plus phosphate is not called nucleoside. Nitrogenous base phosphate is not called nucleoside. Urine or terminal is not a nucleoside. So 16th question, correct option is 1. Nucleoside. Nucleoside is called sugar plus nitrogenous base. Nucleotide. To this base, if phosphate is added, then it is called nucleotide. 17th question. In bDNA, one full turn of helical strand contains bDNA. Generally, DNAs are various types of DNAs commonly located. Okay, so DNA are different types: aDNA, bDNA, cDNA, ddNA, like that. Different types of DNA are there. In that, bDNA contains how many base pairs? 11 base. Generally, 11 base pairs are present in A type of DNA. 8 base pairs are present in D type of DNA. 10 base pairs are present in B type of DNA. 9 base pairs are present in C type of DNA. So in these 4 options, the correct option is B. 17th question, the correct option is third option. Third option is correct option. Next, in a DNA molecule, adenine of one strand base pair with dash on the other strand. In a DNA molecule, adenine of one strand base pairs with adenine always combines with thymine. Adenine always combines with the thymine. Okay, that's all. Thymine. See. 18th question, correct option is 2. Okay. Next, 16th question. Nucleoside is, generally nucleoside is sugar nitrogenous base is called nucleoside. 16th question, correct option is 1. Remaining options are not related. Nucleotide is if phosphate is added to the nucleoside, then it is called nucleoside. In bDNA, one full turn of helical strand contains. Okay, in bDNA, one full turn of helical strand contains bDNA. Okay, next page. In bDNA, the rise per base pair would be, that means what? In bDNA, in between two base pairs, what is the distance? 4.3 angstroms, it is wrong option. 2.4 angstroms, it is wrong option. 3.4 angstroms, it is correct option. 4.2 angstroms, it is wrong option. So in between, adenine is always combines with thymine. So cytosine is always combines with guanine. Okay, so in this case, in between these two, the distance is 3.4 angstroms. Okay, that's all. The correct option for 19th question. 19th question, correct option is 3. Third option is correct option. In bDNA, the rise per base pair would be 3.4 angstroms. Next, 20th question. The nitrogen bases of the two strands of DNA are joined together by phosphodiester bond, hydrogen bond, glycosidic bond, peptide bond, peptide bonds. The nitrogenous bases of the two strands. The nitrogenous bases of the two strands of DNA are joined. In the DNA, nitrogen bases. See, adenine is combined with guanine. Okay, adenine, in between adenine, thymine, 2 hydrogen bonds. In between these two, 3 weak hydrogen bonds. So hydrogen bonds are located. 28th question, the correct option is 2. Okay, see here, the hydrogen bond, the bond formed between two polysaccharide strands of DNA. So hydrogen bonds is correct option. Next, 21 question. The genetic material of tobacco mosaic virus is the tobacco mosaic virus. What is the genetic material? In virus, always genetic material is RNA, ribonucleic acid. Okay, the genetic material of tobacco mosaic virus is single-stranded RNA. The single-stranded RNA is a genetic material of tobacco mosaic virus. Next bit, the double helix model of DNA was proposed by. Double helix model of DNA was proposed by Berzelius. It is wrong option. Watson Crick, it is correct option. Griffith, it is correct option. Raptor, it is wrong option. Robert Brown, it is wrong option. The double helix model of DNA was proposed by Watson and Crick. So 22nd question, correct option is 2. 23rd question. In the 5 prime, see here in these two places, 5 prime end of DNA molecule. So at the 5 prime end of DNA molecule, generally like this way, DNA will be there. Okay. See here, like this way, DNA will be there. Okay. One strand 5 prime to 3 prime. Another is 5 prime to 3 prime. So in this 5 prime end of DNA molecule, what would be there? Okay. See here, the 5th carbon of pyrimidine base is free. It is wrong option. Pyrimidine base is not free. The 5th carbon of purine base is free. It is wrong option. The 5th carbon of pentose sugar is free. 23rd question. Okay. Third option is correct. Pentose sugar of deoxyribose. Pentose sugar of 5th carbon is free. So third option is correct. One end of the strand is called 5 prime end, where the 5th carbon of the pentose sugar is free. And the other is called 3 prime end. So this other is called 3 prime end, where the 3rd carbon of pentose sugar is free. That's why it is called 5 prime end. It is called 3 prime end. So for 23rd question, correct option is 3. 24th question. The pitch of the bDNA is generally the pitch of the bDNA. Pitch of the bDNA means what? See, in single turn, the base pairs is 10 base pairs. So in that 10 base pairs, in between two adjacent base pairs, the distance in between these adjacent base pairs is 3.4 angstroms. In bDNA, 1 turn of a helical strand has 10 nucleotides. 10 nucleotides. The base pairs of the DNA are stocked 3.4 angstrom part. The pitch of the DNA is equal to 3.4 angstroms into 10 is equal to 34 angstroms. 34 angstroms. So 24th question, correct option is third option. So here, second option is wrong. Why wrong means here in one pitch base pairs are 10. In between these two, distance between base pairs is 3.4 angstroms. When it is multiplied, so 34 angstroms. 34 angstroms. So for this 24th question, the correct option is 34 angstroms. 25th, the primary precursor for the production of cholesterol in our body is, see, the primary precursor. Primary precursor means what? So initially immature form of initial product, primary precursor, initial product for the initial material for the production of cholesterol in our body is acetic acid. Acetic acid is a primary precursor. Okay. So in biosynthetic pathway or anabolic pathway, the acetic acid is converted into cholesterol in liver. So option 1 is correct. Citric acid, it is not a citric acid. Ethyl alcohol, not ethanol. So acetic acid, 25th correct option is 1. Acetic acid is correct. The primary precursor for the production of cholesterol in our body is acetic acid. The continuity we will see tomorrow.